And these are going to be the Ascidians, which you can also call star Ascidians for the ones that are colonial. And we're also going to talk about the Cephalo Chordates, which are proper chordates but not quite vertebrates. So they don't have bones, but they have a lot of the other structures. So they're like an invertebrate vertebrate, if that makes any sense at all. So shown here, this is going to be the embryo of one of these. Actually, let's start this. There we go. So we can have the nice little subtitles. This is going to be the embryo of one of these Ascidians. And you can see it's got a little tiny eye in there, and you can see a little pigment at the back of it. That's why eyes look black. You're looking at that melanin pigment. Which we've seen now is going to be common throughout pretty much all the animals, and you can go right back in the fossil record, and it is there, yeah, over and over again. They're going to have that nerve cord and notochord, and they have a little muscular sort of back end so that they can swim through the water. Kind of like an enlarged version of a spermatozoon, really, is the idea behind this body plan. And then they are going to be colonial. So they're going to anchor down, and when they do, then these are going to be individual zooids. So here's one individual, here's another one, here's another one. So they're all going to be the same species. And they're going to cluster together in this sort of arrangement. And so each one is going to have its own little siphon. So they're going to breathe in water right here. There's that little hole in each one of them. And they're all going to line up, and they're going to breathe out their water and their waste from the center. So it's like they have a common latrine or bathroom, something like that, in the middle. Or a communal anus, if you like. Although they actually each have their own individual anus. They just all poop into the same place. And they're also breathing water so that they can exchange gases. So it's a lot of CO2 is going to be coming out there as well. Okay, and so these are going to be in the tunicata, so they are still within the chordates, but they're not vertebrate chordates, and they're not cephalochordates, which are the ones that look like little miniature fish, but without any mineralization, without any hard bones. All right, so what do they have on the inside? So Botrytis is shown here, and everywhere that's gray, this is a creature that does not have the thing. Everywhere that's yellow, it's a creature that does. So let's look at these. Arabidopsis is going to be a plant. Neurospora is going to be a primitive eukaryote. Monocyga brevis is going to be one of those single-celled sponges, the conoflagellates. Amphimedon queenslandica is going to be a sponge. And so you can see these guys have none of these little features. They're pulling out features that kind of distinguish things like chordates from things that are not like chordates. Acropora is going to be a coral. Nematostella is also a cnidarian. Cinerabdidus elegans is going to be a nematode. Drosophila is an arthropod. So you see where we're moving on this family tree. And so we really have nothing there. Even when we get to Strongylocentrotus, this is going to be the purple sea urchin. You can see it doesn't have any of these particular things that they've picked out. But once they get to Branchiostoma, this is going to be the Cephalo Chordate. So now we're on proper chordate track, not just these deuterostomes that include chordates. Let's see. Oikopleura, I'm not sure, but I think that is also a tunicate. Siona is definitely in the tunicata, as is Botrytis. And then, so you can see Botrytis is finally the first one that has all of these little features that we find in most of these higher things. So Petromyzon is going to be a lamprey, so a jawless kind of fish that latches onto other ones. Danio is a little zebrafish, just like the ones in the tank that we keep. Let's see, Xenopus is a frog, Gallus is a chicken, Mus is a mouse, and humans, yeah. And so the genes that we're picking up are involved in a variety of processes. Some of these are actually involved in diseases, and it's not that we picked up the gene in order to have this disease. That is not how evolution works. But these are genes that are doing something, and in a certain condition, when these genes are damaged, then this is the result. And so you can see a lot of these that are involved in cataract formation in the eyes, in deafness, in changes to the immune system. And so, yeah, a lot of the shared equipment that we find in sort of higher vertebrate animals over here everything from petromyzon out to humans, was actually first present in Botrytis. So this Ascidian. So yeah, star Ascidians. Interesting genetically. Okay, and this is showing the early development of the Ascidians. And so here's their little early cleavage. And they have very distinctive appearances in their embryos. They'll have certain cells that will be in certain places. It's not like the chaos that you see with a human embryo, where you've got cells migrating all over the place. Much more tightly regulated, I would say. And you can see the individual cells that make up the embryo. So this individual layer of sort of column-like cells here, these are the ones that are going to mineralize up or harden up with cartilage and actually form that notochord. The dorsal nerve cord is going to be formed on the outside. So these are cells that probably do not know that they're destined to become nerve cells yet, but that's what they're going to become. And then the cells that are going to form the primitive brain will be right up in this area, and the cells that will form all the rest of the organs are going to be down here, but they'll just be stem cells at this point, because there really aren't that many cells. It's not even like you would have one cell per organ necessarily yet at this stage. But you can see the primitive rudiments of a gut forming down in here, and so yeah, that's what we're going to develop down in there. And then this is the sort of mature larva, if that's a thing that we can say. And you can see the nice sort of reddish notochord running down the middle in panel E. And very clearly defined head region and nice little muscles. And the dorsal nerve cord would be running right along the top here at the back. And so this is going to be their metamorphosis. And so we're showing right up in here, this is the part they're going to use to attach down to the floor. So they're going to stick themselves down with that. But the part that's going to form the heart is located right up in here. And it's going to migrate right down to there. Great. The oral siphon is going to be in pink. And so there we go. We've got that up there. Neural complex, this is going to be the equivalent of the brain. And so you can see it's actually coming from where the brain was in the larva. And the rest of the dorsal nerve cord was running down here. There will be parts of that, but it's just kind of like nerves that split out now from this little brain. The rest of the nerve cord is obscure at best, I think you could say. And then the stomach, intestine, and esophagus, this is going to be this sort of blob right here. It has not yet fully developed, but yeah, you can kind of see what's going on here. And then the pharynx, big old pharynx, is right up in this area, and the pharynx is going to be this, it's going to develop from this purple area right here. And so, yeah, that's quite a transformation. That is no less dramatic than the one that humans go through as we go from a little tailed fish into what we are now. Okay, so the last ones we're going to cover are the Cephalo Chordates, and the Cephalo Chordates are going to start out just like a human embryo, and then they're just going to stay that way. They're going to stay small, and they're going to feed with an endostyle for their whole little lives. So the pharynx is going to be in this area. The pharyngeal gill slits are right here. It's these little sort of blue ovals. And that's going to let the water in and out so that they can breathe it. And the body is basically just going to be a kind of like a tube. So this is looking at it from the front end. You can see the brain here and some of those little nerves that are coming out from the brain. The endostyle is going to be this green thing that looks like a crazy glove up here. And it's a little groove lined with cilia that's going to feed material in. And so the mouth is going to be located right behind that. You've got the brain and dorsal nerve cord, but we know we have brains in many other kinds of animals. So it's the fact that all of the nerves coming out of the brain are going to be clustered up in this extension. It's like the brain grows a big long tail, which is our dorsal nerve cord. That's kind of the idea here. The notochord is going to be underneath it. It's this little black rod that you can see curving underneath it. And again, that's not really the place where you would want, if you were laying things out for yourself, you'd say, let's put the protection on the outside, to reduce spinal injuries in the future. But yeah, I don't know. We got to take what we're given. That's just how it works. The anus is going to be number 5, and so this is the going to be the intestine equivalent. The stomach equivalent is going to be up here, and the tail just keeps on going after, and it's got muscles in it, so it can waggle that around and swim, and so that is a postanal tail. And so cephalochordates are going to express all 5 of those traits of the chordates throughout their entire lives, because its embryos are just a smaller version of this, and yeah, they essentially stop. So this idea that your development goes up to a certain stage and then you might just stop or you might stretch it out is a thing called Peta Morphe. And what that means, so that's something that we also see, and I think we might have mentioned it. It's something that we see when comparing humans to chimpanzees. So the developmental program for baby humans and chimpanzees and the way that the babies learn is going to be similar up to a couple months of age, something like 6 months of age. But at that point, the chimpanzee is going to become very quickly sexually mature and ready to fend for itself and hunt for itself. And human babies, I don't know if you've had experience with them, but they are useless at 6 months. They're probably useless at 2 years. Yeah, yeah. Go hunt for your own food, baby. No, that's not going to happen. And so we are Peta Morphe apes, essentially. We are retaining an embryonic state and we're growing larger, but we're not really growing up. Although we do eventually arrive at sexual maturity at what? Our teens, yeah. Much, much later than many other animals that are on this sort of faster development track as a survival advantage, perhaps. So, looking at the genomes of creatures like a cephalochordate and creatures like us, what we can see are, here are the cephalochordate chromosomes. So it has one set of chromosomes. Actually, no, these are the human chromosomes, sorry. The cephalochordate also has chromosomes. Let's jump to the next slide and then we'll come back to this one. So here we go, Branchiostoma floridae. This is going to be one of those cephalochordates. And shown on here, this is the cluster of Wnt genes. You remember Wnt genes are going to be involved in signaling. So on humans, on Branchiostoma, we have one cluster that has the 5 main Wnt genes. On humans, we have 4 different clusters that each contain a minimum of 2 of these Wnt genes and seem to have the same thing coming after them, which is going to be the start of the Hox gene cluster. Whenever there's a little double line like this, that means there are several million bases in between. But yeah, it does appear to be similar. So it's almost as if we took this little cluster, duplicated it into 2, duplicated both of those again into 4, and then through evolution, we start losing some of these genes. So if you were to look in the region in between Wnt10 and Wnt6, I believe you would find a degenerate copy of Wnt3 that doesn't work anymore. But the gene was there. And so this kind of shows that humans have 4 copies of everything that a primitive chordate has. And that's true more widely. So shown here, so we're lining up the human chromosomes against the 17 chromosomes of Branchiostoma up here at the top. And humans will actually have 4 copies of some of these things. And if you look through, so they'll have them lined up 1, 2, 3, 4 like that, when we know that they're 4 separate copies of the same genes. And so from this, we get the idea that throughout chordate evolution, there have been at least 2 gene duplication events. So there are chordates that have exactly 1 copy of everything, which is good and bad. I mean, they have the minimum that they need to get by, but if something happens to one of those genes, if they get a mutation that's picked up in there, then they're just dead. They don't have a backup, nowhere to go from there. And so along the way, the idea is that some of these things duplicated their genes. And I believe you can still find some chordates that have 2 sets of everything. And then once you get to the vertebrates, even the very primitive vertebrates, you're going to have 4 sets of every single gene, like we get in humans, like we get in most of the other vertebrate chordates. And so, yeah, duplication and duplication again, and then wholesale loss of genes. So even though we had 4 copies of everything, we've tended to lose large chunks of some of those. And I think we've ended up with 1 copy of everything we needed, and sometimes 2 or 3 or 4, 2 or 3 backup copies. And these things can now start to evolve on their own. So if you've got one gene that's doing the primary job of keeping the organism alive and serving that basic function, then if we pick up mutations in the other versions of it, then the other versions can start to take on slightly different roles in development or in the body. And so this is the idea behind evolution of the higher chordates like us, or more derived chordates. I guess we're lower, really. We're like a late branch off the tree. We have, multiple copies of genes, and we use them in slightly different ways. So this is the pattern of evolution. You take a gene, you duplicate the gene, and then you start to diverge some of the copies of the gene until they take on other functions. And then bam, now you've got two different functions, and you've evolved in this way. You've got a new gene that started out as an old gene and just changed. It's kind of a gentle way to do evolution, where you can still keep the individual alive while the experimental sort of tinkering phase is going on. And that's going to be the end of this section on chordates. Hope you liked it, and do check out all those videos. There's some really wonderful things in there. These little primitive chordates are quite beautiful, I think. They're some of my favorites. Thank you very much.